The first
Go
question
ahead.
is, what is the difference between the function of neutrophils and macrophage? Because you said that it like neutrophils in blood and macrophages in tissue. So, are there any difference in their function?
Okay, so
Can you
the answer would be yes. So the answer would be they are two different types of cells with very similar functions. And macrophages, they are coming from monocytes, and monocytes are derived from the blood. Okay, so the macrophage is residing inside tissues. So if you have okay, so we start with the monocyte. So it differentiated from the stem cell of the blood, and then it travels through this bloodstream to the place that it should reside in. So let's say it travels to stomach, and it finds a place to stay there. So inside the tissues, it kind of patrols the place. So playing a role of the policeman. And the neutrophils, they are very specific phagocytes. So instead of just patrolling the stuff, and if there is a danger, react on this danger, they try to kind of maintain the attacking power of the organism. Okay, so whenever we have inflammation, all the neutrophils, they fly there and they start digesting everything that they see. Okay, so in this case, they eliminate the pathogens if they receive this information about the inflammation. Okay.
Okay, thank you. And the next question is about the MHCI. As I have understood, they are very individual for every person. And also in the end of the lecture, you said that in the transportation of organs, they try to find the similar MHCI that could be appropriate for the body. So
yes.
Is it like individual as our fingerprints, or there are only several types that could be like variety?
Yes, it is individual. So the person, so even your relatives, they will have very similar MHC1, but it will not be the same. And that's why, for example, when we make the transplantation from one, let's say from the son to mother or from son to father, they still have this rejection of the transplant, because even the most similar ones, they are being recognized as non-self. And in this case, of course, the immune system of the recipient will start fighting the aliens. And in this case, our transplant is the alien, right?
Mm-hmm.
So it is very individual.
Okay. And the last question is about B cells that produce the antibodies, and they like they pass the antibody check, quality check. So is there cases when this check doesn't work and they start to produce like a lot of not appropriate antibodies?
Yes, it is the case in immunodeficiencies, in cancers, and in the autoimmune diseases. So, for example, if the quality check says that we cannot produce this antibody because it can harm the person, so it can harm the cells, the quality check is not passed. So these type of antibodies will be seized, and the plasma cell that produces these antibodies, it will be killed. And for example, in the cancers, they just don't respond to these self-killer signal from the antibody control check. So let's say that they produce the antibodies that can harm other cells, and the quality check says, okay, you shouldn't produce them and you have to die. And in this case, they do not respond to this signal and they still produce all these antibodies, etc. So I don't remember the exact name of this lymphoma, but these lymphomas, they lead to cachexia, which is the running out of the nutrients because everything is being converted into these antibodies. So it's a rare condition. And finally, we have this immunodeficiency because the control of the antibody quality is performed by other immune cells. And if we don't have these immune cells, or we have them in a very low conditions, some of the quality checks, they might be passed without meeting the required conditions. Okay.
Is it connected to disease lupus, or it's different case?
So it might be. So the problem with the lupus is that we don't know the exact mechanism of the pathogenesis. So pathogenesis is the process of the development of the disease, and we know how it develops, but we don't know how it begins. So in this case, we cannot fight it because we don't know how it starts and the early stages of the development. We know how the tissues are harmed and damaged, but this part that is before that is staying unclear. And it might be also because these antibodies, they do not pass the antibody quality check, but still they are being produced. And it's really difficult question because I think the laboratories that work on this lupus, they really want to know what is happening there.
Oh, okay, thank you. It was very informative.
You're welcome.
Hi.
Hello.
Hi. I have several questions. And adding to the previous question that was asked, is there a body that is just unable to produce antibodies?
Yes, of course. So the thing is, all these immunodeficiencies, they usually lead to the people not able to produce these antibodies. So why do these opportunistic infections happen in people with HIV? It's just because usually we produce a lot of antibodies against the main infections. And in these people, they fail to produce these antibodies. So these main infections that will not be encountered in healthy people, they will develop these diseases and usually die because of these diseases. Okay, so it's something like the parasites and fungi in a respiratory system, and many of these AIDS diseased people, they die because of this pneumonia, and usual people will never develop such thing, and it is related to the absence of the antibodies. Okay, so where did I stop and where did I?
You were discussing HIV and fungi.
Okay, so these opportunistic infections and their rise is only related to the absence of antibodies in the organism of the patient. Okay, so they do not produce antibodies at all. Is it what you were asking, or you asked in general the organisms?
I guess there's no there's no disease. I mean, I guess there's no human being that cannot produce any antibodies at all. Is this possible?
Well, I guess it will be fatal if they don't have this gene, because we encounter a lot of these bacteria, a lot of these viruses, and they just die on very early stages of life if they cannot produce them. So even your skin, for example, a lot of antibodies, they keep the barrier even between the human and the beneficial microorganisms, because these beneficial ones, they can harm our body if we don't kind of limit their existence. Okay.
The next question is about lymph nodes. So you said that lymph nodes is an part of our bodies which fight against pathogens, and when they do fight, they get swollen. So
Yes.
when they do get swollen, is it an indicator that it is fighting and we should wait, or is it an indicator that we should maybe take some medications?
So it's not related to taking medications or not taking them, because if your lymph nodes are swollen, it means that the process of antibody production has already started. And it might be the early stages or the late stages, but the swollen one means a lot of blood inside it, and a lot of proteins are being produced there. That's why it needs so much blood and liquids. So it's just like the inflammation drives this swelling of the lymph nodes. Okay, and the need for taking medications is really related to other stuff like high temperatures, for example. If you have temperature that is around 39 and higher, you should probably take these anti-inflammatory medications. And it might happen even if you have the swelling in your nodes. So it's not very much related.
Next question is about natural killer cells. You said something about how they could kill possible or like cancer cells. Is it like legitimate treatment of cancer, injecting natural killer cells?
So we usually don't inject the natural killers. We take the T cells, so the T killers, and we train them. So now we fuse the antibodies against the antigens of the cancers, and we make these T cells express this antibody on their surface. So let's say this trained T cell now can fight the cancer cell because it can recognize the antigens of the cancer cells. So whenever we reinfuse this T cell in the body, so let's say we have one cancerous cell, and this T cell comes to these cancerous cells and senses this antigen here. So we have the reaction between the antigen and the antibody on the surface of the T cell. And because we have this reaction, the T cell starts activating because we have this key lock match, okay? And after that, what happens? So this T cell starts attacking the cancerous cell. So it releases a lot of free radicals inside this cancerous cell, or it sends the signals like you should go into apoptosis, or other stuff that disturbs the life of this cancerous cell. Okay, so we do not use the natural killers, we use the T cells, because the natural killers, we cannot identify them. So they do not express T-specific or B-specific antigens on their surface. But because we could identify the T cells, and specifically the T killers, we can kind of harvest them from blood and then train them, okay?
Is it effective as chemotherapy, or is chemotherapy usually
it's much more effective than the chemotherapy.
Oh, okay. But I haven't really heard about this, which I think it's not really popular.
It's not about popularity, it's about the recent findings. So the thing is, we started training these cells about 5 years ago, because before that we didn't have such technologies that will allow us to train these T cells. Okay, and since it's very new, and all doctors, they are quite conservative, so they don't like the new stuff unless it's proven that it is harmless, etc. And in this case, of course, the doctors, they are trying to get much more information on how harmless it is to inject these trained T cells. Okay.
Okay, so the last question is probably more out of curiosity. Do viruses passively exist in our body, like maybe HIV? Could it be possible?
Again, please.
Do viruses passively exist in our bodies?
Yes, many viruses, they have this persistence. So what they do, they just hide inside the cells. And by hiding, I mean they can both integrate the genome inside the genome of this cell, or just keep their particles in some vacuoles that will not be discarded by this cell. So they persist, and they kind of in a dormant state, which means sleeping like state. Okay, and whenever they have the opportunity to start replicating, they will use this opportunity, and of course the person will develop the infectious disease.
Okay, so I'm asking this because one of my biology teachers told me a story about his friend who donated blood twice in one month, and they found an HIV in his blood. But then, like after a month maybe of taking more vitamins, they couldn't find HIV. So is this like a legitimate thing? Is this possible? So maybe we're treating a lot of
it
the
is
patients
possible,
wrong,
yes,
and just maybe we need to give them time to recover.
So the problem with this HIV is that we really need to remove all the T cells, the damaged T cells, from the body. If we remove it, and we'll be able to replace it with something else, so make the bone marrow transplantation, we could treat the person. But unless we have at least one T cell that is infected with this HIV, it will be impossible for the person to cure from this disease. Okay, and I think that few people, at least two, in recent time, they had this transplantation of the bone marrow, and they actually cured from this HIV.
So HIV could be possibly curable, but not AIDS.
No, they are interrelated. So if we have the HIV, it's not, well, some people, they kind of, they are protected against the AIDS because they do not have these special receptors on their T cells, and the HIV cannot enter the cell. And in this case, the AIDS cannot be developed.
Okay.
So these people, even if they have this HIV, they do not develop the AIDS.
I have a question. Let's say, like, have you heard about the boy in a balloon in 1970s, I think? Like,
is it the guy who was infected with HIV?
no, I think it was a boy who was born with an autoimmune disease. So to save his life, the doctors put him in a sterile balloon. there was a room in a balloon, and he lived there for like 8-10 years before he died. So everything was sterilized, like food, toys. No one touched him directly. Yes, so like that. I have a question. Let's say there is a person with like AIDS, developed AIDS, or other kind of autoimmune disease. Technically, can he be put in such a room and live there for his entire life? Like he can survive, right? Potentially, because there would be no microbes, no bacteria in this kind of sterile environment.
It's true, but it's true partially because if the person does not have the microflora on their skin and in their guts, they will suffer from the vitamin deficiencies, etc. And what else? Well, we technically could do that, but it is related to many stuff that is kind of well, it's a balance, right? So we have scales. On one side we have harm, and on the other side we have benefits, and depending on what outweighs, we decide. So I guess we do not build these boxes, sterile boxes for every single person, just because in some cases it might bring much more harm than the benefit.
Hello, I have a question regarding organs of immune system. So thymus reaches full maturity in childhood, and then it transforms into adipose tissue. So the question is the thymus in adults just an adipose tissue, or how it works like?
So it's mainly adipose tissue, and so we still have the parts of the thymus that are related to the maturation of the T cells and the release of this hormone, and we just don't need it so much because other parts like lymph nodes and spleen, they can take some of the functions on themselves, okay? And that's why the thymus is reduced, so it becomes fats mostly, but still a small part of it might be functioning, okay?
Oh, okay. So like thymus works less efficiently than it worked in the childhood, right? As far as I understood.
Yes.
Oh,
You're
thank
right. You're welcome.
Hello.
Hello.
I have a question about spleen and tonsils. Why would people remove it?
Okay, so people do not remove them on their will. So usually spleen is being removed because it bleeds a lot. So even small heats and small quakes, they can break their parenchyma. So the parenchyma is the functioning part of the organ. And in spleen, it is very fragile. So what happens? So let's say in the car accident, the person had this hit and the parenchyma was ruptured. So instead of, instead of being the whole, it became like this. So and this organ is tightly related to the bloodstream. So a lot of erythrocytes, they are being killed in this place. Why? Because they get older and older. And because we have a huge blood supply of this organ, it starts bleeding. And the thing is, we cannot actively or effectively stop this bleeding by just, tailoring this organ. Okay, so we cannot glue these two parts together again. If it is broken forever. And for the person not to bleed to death, we just cut these blood vessels and remove the organ. And the problem is the person still survives, and the worst consequence might be shortening of their lifespan. So instead of 80 years, they will live 70 years, for example. Is it clear?
Yeah, but what about tonsils?
Oh, yeah, tonsils. So tonsils, they become, so with aging, their function is less and less stable, or how to say that? So other parts of the lymphoid tissues, they take their functions, and many tissues inside these tonsils, so let's say this is tonsil, and many tissues with age is replaced with a connective tissue. So they are not functioning, but a lot of bacteria reside there. So the worst ones are streptococcus, and these streptococcus, it can develop the cross immunization. So we produce the antibodies against this streptococcus, and these antibodies, they can harm the valves of your heart, for example. And in this case, whenever we have very frequent recurring streptococcal infections, we decide to remove these tonsils because other parts can take their function, but we will remove the huge foci of this infection, okay? Yes, it is tonsillitis.
Thank you.
You're welcome.
May I add something?
Yes.
So there are holes on tonsils after the tonsillitis, right? So are these holes curable or what?
Again, please, what do you mean the hole after tonsillitis? After removal of these tonsils?
No, on tonsils there may be some holes. Like
yes, they
can we cure them?
They are already cured because the tissue is replaced with the connective tissues, and that's why actually the replacement happens, because we have constant damaging of these tonsils, and after some time, many parts of them will become the connective tissue. And these holes, if I understood you, so there are conglomerates of bacteria that are whitish, and the doctors can remove them. And after that, especially after tonsillitis, so after the acute inflammation, let's say that this tonsil, it was holding these conglomerate of bacteria, and we removed it. So this thing seems like a hole, but it is already cured because we removed the cause of this inflammation. Okay.
So it's normal, right? Because it is connective tissue. Oh,
yes, it is normal. Again, the problem with these tonsils, if they are replaced with the connective tissues in many parts, and their function is already kind of ceased or very decreased, but they still hold these conglomerates of bacteria, they themselves become the loci or location of the spreading of this inflammation and spreading of this infection. And not to harm the whole body, we just remove it totally, okay? So question from Jandos. When we have high temperature, we feel heat because of high blood flow in the organism. The thing about the high temperature, our body starts producing a lot of energy to supply the antibody production or neutrophil production to kill all these invaders. Third, it is for elimination of the remainings of these invaders, and also for repairment. So these four main reasons why we produce a lot of energy. And of course, many times we produce this energy kind of in advance. So instead of producing, let's say, 1,000 ATP molecules, that will be enough for all these functions, we produce 4 times more. And of course, this energy, since these ATP molecules, they are kind of not very stable, they are being reduced to ADP and AMP, and all this energy is being irradiated, or just a second, I don't remember the exact term from physics. So the heat, this energy is being converted into heat, and it is being spread. And this also leads to this feeling of heat and the rise of temperature. So also why we rise this temperature, because the signals about inflammation come to our medulla oblongata. So it's a stem of our brain. And because the cells of this center, they have receptors to these inflammatory factors, they decide to increase the temperature of the body, because many pathogens, they cannot live at temperatures that are higher than 37 degrees. These all coupled together, it leads to the rise of temperature and this feeling of heat.
I just wanted to ask about antigens, so please correct me if I'm wrong. So when some kind of alien
Again,
cell,
please.
About antigens. So some kind of alien cell enters our body. Our immune system produces antigens in response to attack it. Is it right?
So the aliens, they have antigens on their surface or inside their body, okay? So these antigens, they are special parts, special proteins or carbohydrates, or the complexes of these carbohydrates, lipids, and proteins. So something that can be recognized by something else. And our body produces not antigens, but antibodies. Okay, so these antibodies, they work as the key lock system with these antigens. So let's say this is an antigen, and here we have the antibody. And they match like very efficiently each other. And this antibody has the other side here, which activates the immune system cells. So this part here, whenever we have this matching of antigen and antibody, it changes the conformation and says, okay, so now you have to kill this guy that I attached to, okay? And natural killers or T cells, they start attacking this cell.
So after killing those antigens, what happens to antibodies? Like, do they kind of kill themselves after completing their mission, or freely moving the body? Like,
we maintain the low levels of the antibodies inside the bloodstream, just in case of the other invasion, but mostly we reduce them. And how we do that? By digesting these antibodies. It's simple. So we just reuse the amino acids, and it just happens like that. But we have these memory B cells that will produce a lot of antibodies if we encounter the same infectious agent.
I have another question. It's about how we preserve antibodies. So there are some steps of cleaning and antibody quality check. So what does it include? What's
So,
like the operation?
Yeah, so you obtain the plasma from the donor of these antibodies. And of course, this plasma contains a lot of other proteins, lipids, and even other antibodies. So we have to get rid of them. So how we do that, we have special vessel that has a lot of antigens against these antibody. And we just flow these plasma through this vessel. Okay, so let's say a kind of tube. So let's say this is a tube, and inside on the walls of this tube, we coat the antigens against this antibody, and we make this plasma go through this tube. So all antibodies, they will be trapped by these antigens that are glued to this wall. Okay, and what happens next? We detach. So we assume that in this tube we have only antibodies against these antigen, and then we use special chemicals that will detach these antibodies from the antigens. And in this case, we kind of cleaned the antibody of this specific type to then go to the next step of antibody quality check. So how it happens, we have the reference antibody, so the antibody that is known to be super specific to this antigen, okay? And we compare the results of this pairing of antibody to this antigen from the reference one and from the newly obtained from the plasma. And after comparing the results, we say, okay, so this antibody is specific enough so we can use it, or we say this antibody is not much specific, so we cannot use it, so we discard it. And this is the quality check.
Okay, with high temperature, it's now clear, but what about the low temperature? Why does it happen? I mean, is it due to the lack of something maybe, or how does it work?
It might be the problem of the stem of the brain. So what happens, these neurons inside this center of temperature, they are being confused, and they give they kind of receive the information that the temperature is too high and we need to reduce it. And they send the signal to reduce the temperature production or heat production, and the temperature of our body just drops. So this is the pathological condition. But we might also have it during the day, so it is related to circadian rhythms. So what happens at about 3 to 5 a.m., so when the person sleeps, this temperature center, it is being controlled by other centers, and it needs to reduce the temperature of the body. I don't remember exactly why it happens, so which other stuff is happening while the temperature is low, but this decrease of temperature happens in every single organism, so in every single person, so people. So if you want to know about that, you can go and see why the night shifts of temperature happen. So I guess it's related to the hormones like thyroxine and the sexual hormones. So you should read it, and it's quite nice because of the complexity of this controlling system. Okay, so one center controls the other center controls others, etc., etc. So it's a huge chain of controlling.
I want to add to Jandal's question about temperature. Is it normal that a person has a high temperature, but actually he feels fine?
It usually happens in hyperthyroidism. So the high temperature is very usual for this person, and they get used to it. So they don't feel bad about this huge heat of their body. And usually this person should go to doctor and define the exact cause of this high temperature. So the temperature should be 36.6. Okay?
Okay.
If it is higher, it means something wrong is going on in this body. And it might be, again, the hyperthyroidism, some types of infections. So let's say the tuberculosis, it might raise the temperature, so it will be subfebrile temperatures around 37-ish, and it also needs the treatment, right? And other conditions might bring these high temperatures too. And because it's not too high, many people just get used to it very fast, and they just miss the point when they need the help.
I have heard that if a person is not sick or very rarely gets sick, so it is really bad. Why?
It depends on the person. So if this person exhibits the immune deficiency symptoms, it is bad. But if this person is healthy and has all these good sleeping, nice nutrient consumption, so it's a good diet, also the physical trainings, and like a good mood lifestyle, of course they will not be ill all the time. So their protective mechanisms like immune system, etc., they will be fine, and the infections will be ceased right at the invasion. So it really depends on the person. But if the person is kind of in a hyperdynamic state, so they do not move a lot, they do not eat good things, they drink alcohol, they smoke, etc., and they don't have any disease, it might bring some alertness that, okay, so this guy has something with their immune system.
Mm-hmm. And last question regarding the vaccination. So, do the vaccines protect 100%, and are there any cases with undesirable consequences to the organism?
So it all depends again. So there are countries that produce good products, and usually again we say that it is the North America and Europe. So they have a lot of these quality checks, and of course their vaccines, they are much cleaner and better in terms of the production steps, etc. So they perform all these controls on small groups of people, on small groups of patients, and on cells before all these clinical trials, etc. And in this case, you can kind of trust these companies or these countries and use their vaccine. And most of the time, because they use very advanced techniques and methods to produce these vaccine, their vaccine will give the immunity that is much more stable and much more protective than the vaccine from bad quality countries. And in this case, I cannot say for all of the vaccines, but I can say that some of them, just because the producers or the creators of these vaccines, they were first thinking about money, about reducing the price. So they were using the additional, or the media, etc., that is not very healthy. Of course, these vaccines, they might bring some side effects.
Hi again. So I want to ask maybe like a little fun question. So infants, it's important for infants to get like breast milk and to obtain microbes and bacteria to build their microbiome, right?
Yes.
And do you think about the theory that says that babies tend to put things in their mouths because it's like instinctively they're trying to introduce more viruses and bacteria?
It might be true, but most probably it's related to the curiosity, and since these sensing of the flavors, of the taste, etc., it is one of the most ancient parts of our brain. So it is developed on a very good level. So right from the beginning. And infants, for example, when they hear something or they see something, they still need to kind of associate these visual and audio signals to some kind of behavior. But if they sense the taste of the poison, they will right away understand that, okay, so we could not eat it because it is poisonous. So it might be related to both things, like getting a lot of these bacteria, and to the part of the brain that is the most ancient in our body. If you want, you can read more about it, and I think you might find very good explanatory articles that actually describe in usual words without these hyper scientific stuff. They can describe these concepts of both obtaining the bacteria and obtaining information through the taste.